hold your applause for Dr. Sheline. I will make a very brief introduction about her. So Dr. Sheline was recruited to Penn in 2013 as director of the Center for Neuromodulation in Depression and Stress and McClure Professor of Psychiatry, Radiology, and Neurology. Dr. Schilling is known for her pioneering work, widely cited in the psychiatric literature, on hippocampal volumes loss in major depression, as well as the modulating effects of antidepressant treatment. Her research integrates structural and functional neuroimaging in the course of depression, explores neuropsychological correlates and treatment outcomes to determine how depression affects the brain, and evaluates how stress produces functional dysregulation. And this novel work investigates neuroimaging treatment effects of antidepressants, cognitive behavioral therapy, transcranial magnetic stimulation, and real-time fMRI feedback. Dr. Schilling has received numerous awards and honors for her research, has served on and chaired numerous NIH review committees, and she has an extensive track record mentoring junior investigators. She has served on the board of directors for the American Association for Geriatric Psychiatry, board of directors for the Advanced Research Institute, American Association for Geriatric Psychiatry, and she is an elective fellow in the American College of Neuropsychopharmacology. Let's welcome her.
Thank you very much. Tonight we're going to talk a little bit about the brain consequences of depression, what they are, what the mechanisms are, and what we can do to prevent it. So as you heard from Cedric already, one of the things that a lot of people don't realize is that major depression is the leading cause of disability worldwide. Let's see here. As he was saying, partly because it's so prevalent, but also because it causes profound disruption in people's lives, making them unable to function as well in the workplace. About 50% or slightly less than 50% of patients remit with treatment. So unfortunately, that leaves a lot of people who are being treated but really are not able to fully recover. So it's incumbent on us to develop new treatments that will be more effective, to improve the treatments that we already have, and to target treatments specifically for specific patients that will be the most effective for them. So what do I mean by brain damage from depression? I don't mean the massive kind of trauma that you would get from a traumatic brain injury, and I don't mean a discrete kind of lesion like you would see in a stroke here, where you can see this very discrete lesion. It's something more subtle, and in order to understand that, we have to really look at translational neuroscience models. One of the ways we can do this is by looking at rodent models, which were developed in the 60s, and there are various ways you can stress a rodent. You can use restraint stress. You can use an intruder model where a very mean, aggressive mouse is put in the cage with another mouse and beats up on it repeatedly. Or you can do different kinds of conditions in which the mouse gradually gives up. And what you end up with is a mouse that's not so different from a depressed human, although some fundamental differences, but in which you see much less locomotor activity. You see cowering in the corner of the cage. You see less of a desire to eat, and especially rewards like sucrose. So yes, you can get this behavior either by these different kinds of behavioral paradigms, or you can actually inject mouse stress hormone, corticosterone, into the mouse, and it has the same kinds of effects as if you had done a behavioral paradigm to induce the stress. When you do that, what you see is damage to this important structure here, the hippocampus, which this is shown in a human, but it's pretty similar in a mouse as well. And this is the major structure that gets damaged with repeated episodes of stress. And you can see this under the microscope. This is from some work in the labs of Bruce McEwen and Bob Sapolsky, showing here on the left, this is an intact neuron with stress, repeated episodes of stress. You can see these apical dendrites retract because they're damaged. And then if the mouse is allowed to recover, these apical dendrites grow back and you have the intact neuron again. If the stress is not too prolonged or too severe. About a decade later, what was shown here are these spiny processes, that the structural damage is not just in the neuron itself, but also in the synaptic connections, so that when you have an episode of stress or depression, you get less of these spiny processes and less synapses and communication among nerve cells. So as you could imagine, that would help to explain some of why when we're having a severe depression, we are not really thinking as clearly as when we're not. So we were very intrigued by these studies, and this was in the early 90s. MRI imaging had just advanced to the point where we had 1.5 Tesla scanners, which was miraculous. You could actually image smaller things at that point. And so I was thinking, well, if this were true in mice, what would it be like in depression? If we could image the human hippocampus in depression, would we be able to see something that was different about the hippocampus? And so I did that. I recruited people with recurrent depression who were otherwise physically healthy but had had multiple episodes of depression. And we showed that compared with controls, you can see here that the people with depression had smaller hippocampal volumes. And I want to point out, of course, that this is not a diagnostic test. There's complete overlap in these points between controls and depressed people, but it's on average a smaller hippocampal volume. So this is the first time that that finding had been published in the literature, and since then it's been replicated over 100 times. We then thought, well, what about treatment? Can that change what the effect of depression is? And so we first characterized what the relationship was with the amount of depression that people had had over their lifespan and the hippocampal volumes. And you can see here that the more depression people had had, the smaller the volumes. But what about treatment? What if the person had been treated for some or all of those episodes? Or what if they had never been treated? So we brought everybody back and went through a fairly laborious process of really characterizing exactly what treatment had occurred during each of the different depressions that people had had. And when we did that, what we found was that when people thought about the untreated episodes, they had an even more significant relationship between the total lifetime duration of depression and hippocampal volumes, whereas the untreated, and when the treated episodes were counted up. And by that I meant just antidepressant treatment. We didn't count psychotherapy in treatment. Then there was not this relationship. So that's an example, a kind of exciting example that there is something you can do about having depression, which is you can protect yourself from at least some of the effects by having antidepressant treatment. Other interventions also can change the response of the hippocampus to stressors. One of those is exercise. This is from the lab of Rusty Gage at the Salk Institute. So on the left you can see these nerve cells in green, hippocampal nerve cells. And then in the interim between A and B, what the mice got to do was to run on a running wheel. And mice really like to do this. So they were in their cages running away. And then they were imaged again. And you can see here in red these newly born neurons. And this process is called neurogenesis. So the ability of the brain to generate new neurons over the lifespan, which is true in humans as well as in rodents. If you count up the amount of running that mice do when they're allowed to run, It's the equivalent of about 26 miles a day. Fortunately, fortunately, this has been looked at, and you don't have to run a marathon every day to get the benefits of exercise. But just to compare, this is, if you were a mouse, this is how much running you'd be doing. Another thing that we've looked at more recently is a kind of treatment called cognitive behavioral therapy. It's a talking therapy, but it's very routinized, it's standardized, it's literally by the book, because for each session, the therapist gives the person a specific set of techniques to reassess how they view themselves in the world. So instead of focusing so much on the negative, they can think about other ways in which to view themselves so that they're not beating themselves up constantly and not thinking the world is a terrible place. When we do that, what we see is that people change in the way they respond to stimuli. I'm not showing you pictures here about the amygdala, but one of the things that we see characteristically in depression is a focus on the negative. And specifically, when you give someone a negative stimulus, such as a fearful face, the amygdala lights up much more in depression than in somebody who's not depressed. Likewise, the prefrontal cortex, this thinking part of the brain that is sometimes called the executive region, or the region with cognitive control, is less active in major depression. So what we did was showed people these kinds of stimuli here. The task was to match the houses, but at the same time they got exposed to either fearful faces or neutral faces. And I'm not showing you the response from the amygdala, but it was hyperactive. And then with treatment, with cognitive behavioral therapy, it returned to normal. What I'm going to show you next is that the prefrontal cortex, this inferior frontal gyrus region here, did increase. This is the difference between before and after treatment, did increase in its activity as the person was able to improve in their depression outcome. And now I'm going to show you the same region, this prefrontal cortex region here and here, that I had showed you in the previous picture, which is part of a brain network that is involved in cognitive control. We'll come back to this slide later because we'll be talking about connections within and between networks. So another way that we can characterize whether the brain is doing things differently before and after treatment is to look at how, as Cedric was alluding to, how connected it is within a certain region and between that region and other regions. And so when we show this prefrontal cortex region here in blue, this is just showing where it is, and then we look before and after treatment at how connected the amygdala region, this overactive, fearful region, is with the prefrontal cortex. If you treat somebody with CBT, you get, again, in the same region, an increase in the connectivity between the amygdala and the prefrontal cortex. So you get, again, a situation where there's more balance between those regions, and the cognitive control of the prefrontal cortex keeps the amygdala under wraps, so to speak. And you can see that here. Here are the patients before treatment. They have less connectivity, and then after treatment, the connectivity returns to normal. This is a group of patients made up of major depression and post-traumatic stress disorder, both of whom have a degree of depression. And that's kind of sneaking in this idea that depression is a dimension. This is the direction that the NIH is going right now, which is that we really need to look across disorders and think about different symptoms as part of the disorder. And what it suggests is that there's a continuum of severity of symptoms that can be associated with severity of dysfunction in particular brain networks. And it's relevant to translational science, neuroscience, so that we can think about humans and other species and what those dimensions are. Just to illustrate it again, if you have people with a bunch of different symptoms, you can characterize them as having major depression or PTSD, or you can pull them apart by the symptoms they have, such as depression or anxiety or cognitive control. Oops, sorry. And we're actually trying to look at that more carefully in a sample of patients that we're collecting right now, where we're getting imaging information as well as clinical measures, and we're even getting wearable devices. So like the mice, we'll know how much people move around, how much they sleep, what the diurnal variation is. That data will probably be available to you in a year or so, but we don't have any of that data now. But we do have some data on looking at network models of depression in a large multi-site study of data collected across the country, where we looked at brain measures in people with depression. These are the different networks we saw before. Here's our friend, the prefrontal cortex. And we also looked at how connected these networks were within the network and between the networks. And what you can see here, which I'm not going to explain, but just to say that some networks were overly connected within the networks or were overly connected between the networks, and some were under-connected. So here's the frontoparietal network, the one we've been looking at before, and you can see in blue it's underactive, under-connected. What we then did was looked at the symptoms that people had, these symptoms of anxiety, of depression, how much suicidal thinking people had, over 200 different symptoms. And we used a technique to cluster the symptoms and correlate those with these network abnormalities. So here are the network abnormalities shown. Here are the ones that reach significance. And when we correlated that with the symptoms, you can see that by far the biggest correlation was with physical and emotional abuse and neglect. We were very surprised by that. We had thought that the so-called anxious misery symptoms here would be the big correlation that we would see with abnormal networks, because that had all of the suicidal thinking items, it had anxiety, it had depression, and so forth. And yet what we saw was that even 30 or 40 years later, the scar of the early emotional abuse and neglect was what we saw driving the network abnormalities. And it reminds me of a classic study in the 1950s where monkeys were removed from their mothers, and these monkeys grew up to be terribly, just like completely incapable of living in monkey society. They didn't function very well. So Harlow had the idea that you could look at wire mother monkeys and cloth mother monkeys as surrogates. And at that time, the idea was a behavioral one, which was that the bonding that occurred was due to need satisfaction. So if that were correct, then if you attached a bottle here to give milk, then the monkey should bond with this wire monkey. But that isn't what happened. The monkeys were clever. They would come over, drink the milk, and then they'd go back and snuggle with the cloth mother monkey. You can see here. And so it was a groundbreaking paradigm shift where people now started to think about what is the biology of attachment, and has become an important area in neuroscience. So I'll end with talking about the kinds of studies we have going on in the lab right now. One is a computer-augmented form of CBT. We're also looking at novel pharmacotherapies, including using anti-inflammatory agents as augmenting antidepressants. You'll be hearing more from Dr. Othes about transcranial magnetic stimulation, but we're also adding cognitive tasks to see if we can enhance that. And finally, real-time fMRI as a treatment for depression. Things we aren't doing yet but would like to do in the future are looking at more focal treatments such as focal ECT or magnetic seizure therapy or focal ultrasound. But ultimately, what we are aiming for is to develop personalized treatment where we match people up to the particular treatment that's going to be optimum for getting them in their optimum antidepressed state. And that's the goal of our center. So I will stop there and thank the wonderful group of people that I work with in the CNDS and take any questions. Yes.
Have you been able to compare different forms of psychotherapy other than cognitive?
I haven't tried, and I'll tell you why I probably won't. As I was saying, CBT is by the book, it's manualized, and one of the, probably the biggest problem we face in doing comparative research is having treatments that are standardized enough that you're not swamped out by the variability across things. So in regular therapy, talk therapy, each therapist has a slightly different way that they're doing it. And I think to try and see what the effect of the therapy per se was versus the effect of the therapist would be very hard. Yes? Cedric, yeah. Mm-hmm. Well, there are actually a number of other, well, first of all, I have to admit that mice don't kill themselves, and so that probably won't cover the suicidal aspect of depression as a model. But a number of other animal models do use non-human primates, and I think that's one way in which we can kind of extend the models a little bit further in translational neuroscience, because we can use these primates that have a cortex that's much more similar to the human cortex. But yeah, I agree. I mean, the problem with research that has been done to date is we don't have any means so far, other than actually a glimmer of hope with some of the newest pharmacotherapies. I don't know if you know about ketamine, but that's a very rapidly acting antidepressant and has been shown to have fairly good anti-suicidal efficacy. Yes?
Is anything known about the mechanism behind the damage to the brain in depressed people? Why the neurons are damaged?
So I think that was one of the points of the talk, was that the stress that causes structural changes in the hippocampal neurons is one of those reasons.
But specifically, how does some consequent stress actually cause damage?
Okay, well, I don't know if we want to get into this here, but the mechanisms involve glutamatergic cellular actions that then cause a calcium influx into the cell, which is the ultimate endpoint. Yes?
So, maybe this is a naive question. The depression is caused by the changes in the brain, or what comes first?
Chicken or the egg? So it's very multifactorial. Certainly there are genetic predispositions to depression. Depression is a familial disorder, so there is that as kind of the environmental thing. I think I showed you pretty powerful evidence that childhood circumstances such as abuse or neglect also can play a pivotal role. And I think if somebody is depressed, they also, through the stress mechanisms, are much more susceptible to other kinds of illnesses, such as we know there's a huge increase in cardiovascular illness and even in dementia. So I think it's as you start to tease apart the mechanisms, you find that there are some common mechanisms that cross multiple different disorders.
Can somebody just get depressed without having some external event?
You mean like out of the blue? Mm-hmm. Yes, absolutely. I mean, in about 50% of cases, there is an external stressor that can be identified, but the other half seem to be more nearly out of the blue, as near as the person can tell. Sure. Up here.
Describe the stabilizing effect on hippocampal volume in regards to use of antidepressants. Is that stabilizing the size of the hippocampus correlative with ability or reduction in disability over time?
That's a very interesting question. I don't think anybody's really ever looked at that, but that would be interesting to look at. Yes?
I'm not sure if this is the right place for this question, but maybe you know the right place to direct me. It's clear from the slides we saw at the beginning what a huge negative impact mental illness has on people's livelihoods, et cetera. Yet insurance coverage for this treatment is absolutely atrocious. How can that be changed? Or where are resources to lobby, et cetera, for that to change?
I think that's going to have a political solution that many of us have been looking for a very long time. And, you know, if you can solve that one, you could be a really good president. Okay, thank you. Sure.